was just going to start from the beginning. Well, it's a good place to start. So fundamentally, when we're thinking about working with biology, then we need to be thinking about how do we want to approach the science that we're doing. And fundamentally, biological research is hard. It'll always be hard because as we make progress, we're always just going to be going for ever more complex problems to solve, because that seems to be what we're like. We're somewhat masochistic in that regard. And experiments are always going to be costly, right? Irrespective of the experiment you're running, it's always going to be way more way more costly to do, say, an experiment than some kind of in silico analysis or whatever, right? The moment you hit the lab, you're going to be spending money. So the onus is on us really to think, how should we experiment, right? So it's taking our thinking maybe a level higher than the usual thing. When we have a problem that's presented to us in the lab, we're thinking about how to solve that problem, then we maybe don't think very much about, okay, what approach are we going to take to experimentation? And really, we need to be thinking about this in particular because of the system we're working with. Biology is not the same as other sciences. Biology is a system that is truly more complex than anything else. It is the most complex system in the known universe, if we're going to talk in grand terms, right? And most specifically, biology is emergent. What do I mean by that? So an emergent system is something where you have lots of components to it, which then come together in some unpredictable way to then manifest the phenomenon that we then observe, right? So if we think about this is present at pretty much every level of biology, all the way from probably the highest level, which is consciousness, all the way down to the way that an amino acid sequence will emerge into a particular structure and function of a protein, right? It's all got a hell of a lot of unpredictability to it because of these unpredictable interactions between the different components of the system. This is something that is worth bearing in mind now as we go through thinking about how we're going to do experimentation. And just pausing for a second, like Rob and I were chatting before this, our objective here with all you guys in the room is not to convince you fully of, that you should be using DOE or these kind of methods. I would be very happy if we get to the end of this and make you just think, this is something we need to look into more, right? Because I know from experience that the conviction point comes with running your first successful DOE, and then you're hooked. I can guarantee it, right? But there's a lot of kind of things between first hearing about it to actually running that first DOE, and lots of barriers that you have to overcome and lots of kind of questions and hang-ups. And I know because I went through them, and I've also then over the many years that I've been giving talks like this, seen those hang-ups in others, right? So let's just have a think about what it looks like to be doing science in the way that we are all taught to do, right? So certainly in the UK, then when you're first introduced to the scientific method, you're said, look, life is complex, nature is complex. So if you want to understand what effect a particular parameter is having, you have to fix everything else. Otherwise, you'll never be able to deconvolute which parameter is actually having the effect on your system. So this is called one factor at a time, or OFAT, right? So let's have a go through a thought experiment. So we can pick our first factor. We think, we've got a system that we want to optimize, and we use our experience to think, what's likely to be the most important factor that we need to think about? It's often temperature. Temperature is an important factor. We then select a range that we want to look that factor over, and we set certain set points in that range that we want to look at. Okay, so in these diagrams, each dot is another experimental condition. Okay, and then you go into the lab, you run tests, you hopefully do it in triplicate or whatever, and you take averages and you plot them on a graph and you draw a line through them, because this is what we do, right? And then you're like, okay, so our optimum temperature is 25 degrees, but that isn't going to be the only parameter that's going to affect my system. I know there's more. What other parameters could there be? Oh, well, maybe pH, right? And so we'll look at pH as well, and we'll do it at 25 degrees because that was our best temperature, and we'll find the optimal pH and we'll say, okay, this is our optimum. Now, I've drawn the graph in this way for a reason. If you had it in an academic paper, you'd never see a graph like this, right? If you had to look at temperature and pH, then there'd be two graphs next to each other, right? One for temperature, one for pH, and then in the legend, it would tell you the settings of the other factors if they're being helpful. And what that hides is the fact that by doing experimentation this way, we've just taken two cross sections through a multi-dimensional space. And the issue is that by doing that, we've missed something very important. And these results look like they've given us an optimum, but they would also be entirely consistent with if the response surface that we're modeling looks something like this, and our optimum actually lies somewhere across the side. And the simple thing is we just don't know, right? From the tests we've done, we just don't know. So this is an interaction, right? It's where the optimum of one parameter depends on the level of another parameter. Let's go back to what we were talking about earlier about the nature of biology. Like, one of the core features of the system that we work with day in, day out, that we battle to make work day in, day out, is that it is emergent, that these interactions are all over the place. It's almost like a defining feature of biology. And yet the way that we do experiments routinely completely misses these interactions. So what's the alternative? So I have a bit of a love-hate relationship with the term design of experiments, because everyone designs their experiments. Even if they're poor designs, they still design their experiments, right? So I'm going to be referring to it as multifactorial experimentation. There's all sorts of ways that you'll hear it referred to, high-dimensional experimentation, so on. So what it can look like is, okay, we know we want to look at many factors simultaneously, and we want to see how these factors interplay with each other. So then, instead of just doing straight lines through design space, what if we set up a multi-dimensional matrix of conditions within that space? And so often, whenever we're talking about multifactorial experimentation, people always put a cube on the screen. It's traditional. So we'd look at like temperature, time, and pH, and then for each of these factors, we're looking at high levels for them and low levels of them, and all the combinations thereof, right? And by looking at all those combinations, we can then start to look at the system in a holistic way instead of in a very reductionist way. We look at levels for these, and if we want to see, okay, what effect temperature is having, then actually it's really quite simple. I think DOE can be quite intimidating to a lot of people, but like at its heart, it's a pretty simple thing. You take an average of the data on one side of the cube, and you compare it with the average of the data on the other side of the cube. That's basically what DOE is doing under the hood. It's just doing it with many, many dimensions simultaneously. That's where it gets more complex. But it's essentially just simple linear interpolation. This is not like rocket science, right? It's just a much better approach to deal with the system that we have. And the real beauty of it is that actually, if we want to see what effect temperature has, in this case, if we leave it for 3 hours or overnight, then we can see how the things change as you look at a change across the top of the cube versus against the bottom. And then we can see that interaction between temperature and time, right? Very simple example. And it is very simple. So we know that actually biology isn't that simple, and there'll always be more than 3 parameters that we might want to look at. So what does it look like when we actually add more dimensions to the space? So unfortunately, it's very hard to visualize 6-dimensional space on a 2-dimensional slide. But this is my best shot at it. And don't worry if it doesn't make perfect sense of my trying to add more dimensions and more dimensions. But the point of this slide is that all of a sudden, by adding 3 more dimensions to it, we've got an explosion in the number of experimental runs that we'd have to carry out. So where you're looking at 2 levels for any given parameter, it means that the number of experiments you have to run is 2 to the n, where n is the number of parameters that you're looking at. So, you go from 6 to 7 parameters, and you haven't just increased the number of runs a bit, you've doubled them. And so this is an exponential explosion, and then all of a sudden it seems completely unfeasible once you get past a certain number of factors to go any further. But this is where the beauty of this sort of area of experimental design comes in, because you don't actually have to run all of these runs to get the main information that you're worried about. Like, actually, it would be a massive surfeit of runs in a lot of cases. You just don't need them. what these experimental designs do is they very carefully and very systematically take away a number of those experimental design points such that you can actually have a much more efficient experiment that still allows you to see the main things that are going on in your system and to see how these things interact with each other, right? So, and if you think about it another way, because this could be a scary thing, like, hang on, you just, we're not doing all the combinations anymore. I thought that's what this is all about. But if you think about it, in this particular instance, we're still looking at 16 data points versus another 16 data points, right? How often do you replicate 16 times, right? Not so often, right? So there's still actually a lot of statistical power, even though we paired away a lot of the design points. And there's lots of technical reasons why this works, but I'm looking to give you kind of an overview as we go, and to, like I say, just spark that bit of interest and that bit of curiosity about, oh, hang on a second, maybe the way that, the typical way that people are doing biology is underpowered. And I'd say, now we just have a huge abundance of evidence that working this way is just dramatically better. Okay, so I'm going to go through an example. I'm going to speed up a little bit. So the DOE process is actually quite similar to the process you'll already be doing in terms of that scientific process you're going through. It's just where maybe we're thinking about the process we're working with to a greater level of depth and granularity when we're planning, and we're prioritizing those factors that we want to look at. So first off, we need to think about, okay, what factors, so what parameters do we want to test, and what levels, what settings for those parameters, right? And what we need to be thinking about there is, what do we already know? Because we'll know a fair bit, hopefully, about this system already, and there's no point us doing experiments to show us stuff that we already know, right? And then what do we want to find out? And ideally then, in stage 2, we want to think about an experimental design that addresses what we want to find out whilst making the most of what we already know as much as possible, because then we're going to be as efficient as we can in our experimental design. And when we're looking at that experimental design, we have to do some really critical things, like how much resource do we actually have for this experiment? As you'll see in examples through these decks, actually, it's quite nice, we were going through them before. We've got examples from ones where there's 1,200 runs, big run, so pretty big, punchy experiments. Actually, there's one with 1536, all the way down to 33. So if you've got that critically expensive reagent and you really can't run that many runs, this will still work, and it will make the most out of every single one of those runs. If, on the other hand, you can run a full 1536 well plate, well, hell, you're going to get to your answer very, very quickly and definitively. So it's really flexible in that way. And then, when you've got your experimental design, then you have to actually get into the lab and run it. And this is often complex and it's hard. And, quick plug, this is where Synthace is focused, right? We're all about taking that experimental design, helping you to get to that experimental design, and then just working out with the tedious detail of how you set up those 1536 runs where you've got 10 factors that are changing in every single one of those wells. Very, very complex, but actually it's relatively simple if you have the right software to help you, and then the right automation, like the dispensers that we work with, that can then actually carry out that experiment for you. So when it's run, then you have to get the data, you have to structure it, you have to put it back into the context of the experiment, and then you can build models. And all the model is it's nothing, again, it's nothing mysterious. It's basically just saying, okay, if we mathematically analyze what's going on with these data, what seems to be the patterns in them? And what's the statistical evidence that those patterns are real? And then, something that I think is really important, you should then actually look at those models and think about what they actually mean, right? And put it into words. Like, as a biologist, I now understand this about my biology that I didn't understand before, right? Neither Rob or I are statisticians, right? We're just very passionate biologists who know that this is a way that you can actually cut through biological complexity better. So we've got an example here, which is the expression of a human drug metabolism enzyme in E. coli. It's from the early days of Synthace. It's still one of my favorite examples. And there was some literature precedent for this, but the yields were really poor. And what I'm showing here is the iterative work that we did to navigate our way through this complex design space to get to an optimum, right? So first off, we're just doing an experiment where we're looking at which factors are important. We looked at 15 different factors, and it's like, when you're designing it, you don't know which ones are going to turn out to be the important ones. And so you run an experiment to find out. And then we had to iterate, and then you kind of refine and you find, and all the time you're shifting and you're navigating your way through the design space to try and get to the optimum, right? So this first experiment, we're doing 15 factors, and interestingly, we were looking at genetic factors alongside process factors. So genetic factors like, the different parts of the plasmid construct that we're using, so plasmid copy number or ribosome binding site strength or whatever, and we were then looking at those systematically in the context of different expression conditions as well. So this is kind of a protein sciences example. And these are the results that we got from that first week, right? So we ran 64 experiments looking at 15 factors. So it's a sort of very lean experiment. And a lot of these were zeros. And the guy who was running it in the lab came to me and he said, "Oh, Markus, it hasn't really worked." Because he was looking at the zeros, and I looked at the data and I thought, this is perfect, this is exactly what we need. Because actually, this kind of experimental design makes as much use of the zeros as it does of the actual points that they actually work. And you stick it through the analysis, and I'm not going to go through how these plots are generated, but suffice to say, everything on this line is noise, right? And everything off the line are parameters that are having an effect on your system. And I won't say could be having an effect on your system. This is with so much statistical power, you get definitive results. And this is the reason that people like me got hooked in the first place on working this way. Because all of a sudden, like, we're going from a place where it's so common to have ambiguous results, to be not sure exactly how our results from that week's worth of experimentation actually pertain to our problem or not. When we get clear results, it's a real cause for celebration. But with DOE, it's just normal. That's what you get, because you're running an experiment which is actually commensurate with the complexity and the nature of biology. That's fundamentally what it is. So here, we found that the co-expression of chaperones was really important and leaving it for enough time. So this makes sense. We're expressing a mammalian protein in E. coli, and often it'll take a bit of folding, it'll take a bit of coaxing to get where it goes. So it makes sense to us as biologists. And then we also see that there's a synergy, right? It isn't just like, okay, leave it for some time, enough time, and we have chaperones, and that's additive. It's actually multiplicative, right? It's, 2 and 2 is 5, right? Main conclusion only works when you have the correct string and you give enough time. Right. So then what you do is you make sure you use chaperones and you give it enough time. So you've shifted the queue to a place where the model has predicted it should be better, right? And sure enough, all the results go up. It's like, it's almost like cheating. All of a sudden, we're just making day-to-day progress in biology where we're just not used to it, right? It's brilliant. Anyway, so then that gives the model a lot more to get into, and you see all sorts of other stuff popping out. And so, you can then look at all of those, and again, we can see a slope, so we'll move up the slope. We shift the cube again. And at this point, we've been testing 15 factors up to this point, but 8 of those factors have done nothing, right? The high point, the low point, it's been flat. So why bother keeping investigating, right? We put all of our energy into those remaining 7 factors. And once again, it goes up. But now you'll see something interesting, because the average has gone up, but we've got zeros again. And I thought we had left zeros behind. And this is where it gets really interesting, because what we see here is an example of a 3-factor interaction, right? So we've already seen a 2-factor interaction, the synergy that we saw in experiment 1. Now this is where it gets deeper, right? And so we can see the 2-factor interaction here between temperature and time, right? So most of them are all about the same level, and you can't see my cursor, but they're all about 0.4, except if you leave it for a long time and you have a low temperature and the plasmid copy number is low, then you have an opportunity to get twice the active U. It's nice. But then what about when the plasmid copy number is high? The nature of that 2-factor interaction changes entirely, right? So again, most of the conditions still about 0.4 units per mil, but there's also a combination of conditions where everything falls off a cliff and you get zeros. And we call this a design cliff in Synthace. And I'm convinced that this is the source of so much variability and heartache across biology, is that often we're on the edge of a design cliff and we just don't know it because we haven't done this kind of experiment, right? So the PCR that worked one day but then didn't the next, right? You're on a design cliff. So in this case, if you have the temperature too high, you have the plasmid copy number high, and you don't leave it for very long, so it's basically just ramping everything up to 11 and expecting it to work, then it just falls off completely and you get zeros. So the nice thing about here is that then we know how to make sure we never get zeros, and that's by basically by using low plasmid copy number. So long as we use the right construct, we're never going to be anywhere near that space. And so by understanding the system, we can build in robustness, right? It's not just about something that works, but it's something that's going to work every single time. Anyway, so we iterate it once more, and up it goes again, right? You'll see a pattern here, just every time. So this is 4 weeks of work. Week 1, week 2, week 3, week 4. I was quite happy in week 1. And look how small those bars look like now. I mean, I tell you, there's nothing like making this kind of progress so definitively within biology. It's awesome. Anyway, then we tested, and we made sure that actually the design points would predict to consistently give good yields. We scattered around in that design space. They did give consistently good yields. It's always worth testing the model, right? Instead of just, believing it blind. But anyway, so this is still my favorite case study. Hopefully it gives you a bit of a walkthrough why multi-dimensional methods can be so powerful. So let's look at some case studies that aren't from Synthace. So a couple of years ago now, Helena from AstraZeneca stood up on stage at SLAS Europe and gave a presentation about what they've been doing with Synthace and with high-dimensional experimentation in their labs. And I've just got a few screenshots from that presentation to give you an idea. So this is where they were looking at 9 different factors simultaneously for a buffer optimization for a biochemical assay. And they wanted to look at, can we change the KD of this system? And the answer turned out to be no, right? But because they'd run 75 different conditions and run full dilution curves in each of those conditions, so it's actually 1,200 runs, a very, very complex experiment, but something where Synthace was doing all the heavy lifting for actually planning and running that experiment for them, then you can then get to a definitive answer of no, right? So instead of going round and round and round circles, testing another factor and another factor and another factor, hoping that maybe you'll get there, you get the bad news early and you can move on, right? And your stakeholders get that news clearly as well. So they can't go back and say, oh, but have you tried? Because you're like, yes, we have tried. We tried all of it. Now let's move on, right? This is an example where, because you're mapping out the whole space, so this is a type of design called space fill design. So on the left-hand side of the slide, instead of the cube, you could see like there's a cloud of design points across the design space, like which has filled up the whole design space with a load of different points, all kind of like evenly spread throughout that space. So it's a different type of design. It's the only kind, you'd only ever want to do this with automation, right? This is run on a SPT Dragonfly. And it means that then the combination of that software and the automation means that you can actually just almost, you can set up any arbitrary set of conditions you want, right? And we do that all in parallel, fills up the space. And so it means that not only can you look for where the assay window is good, but you can also see where it's cheapest to run that assay at the same time. So you can opportunistically take a massive cost saving at the same time as having a really high quality assay. Another example, this is done on a 1536 well plate, straight into a 1536 well plate, and it was a doubling of the assay window and a significant improvement of the Z prime, all in a single experiment, right? So in all of these cases, it's going from a system where, we have some ideas about how it might work to complete understanding of that system in a single experiment. And that's what this is offering, and that's why you see such dramatic effects. And we also have a couple of case studies from GSK. So these are both case studies from placement students at GSK, and I think this is a really interesting thing in its own right. So these are undergraduate students who spent a year at GSK to, learn what it's like to be in industry. Now, when I was one of those students, I got nothing done in that year, right? I mean, I'd love to say that I did, but science is hard, right? These guys, they did 10 assay developments each in that time, undergraduates, right? And again, it's because it's a very powerful method that just delivers the results really quickly. Again, using Synthace, combination with Dragonfly, and then producing these high-dimensional data. And Lizzie in particular, I'm really grateful to, because she also put the impact on her poster. So all I have to do is copy and paste and put it onto the slide, right? So, and it's getting very normal now to see these kind of results. It's not like 10% more efficient or 20% more efficient. It's common to see 85% reduction in the number of experiments, 20-fold cost savings. And it's because you're mapping the whole experiment with a single run. Like I say, we're now seeing a really big and overwhelming buildup of evidence that this is just a dramatically better way of doing this kind of very tough science, where you have to do different science every time a new program comes through. It's the kind of area which has been stubbornly difficult to industrialize. There are areas of discovery where we could just make things high throughput and we can put them on some beautiful automation that's down in the exhibit hall and we can crank it through. right? Whether it's like, CRISPR array screening, or like a high throughput screening, obviously in hit identification, or like high throughput omics. Like these are all things that we can standardize and we can crank for a large part. But then there's all these areas which are just a hell of a lot harder than that, where you've got teams that are under huge amount of pressure to get really difficult science done every time. And like Rob said, it's indeterminate, it takes a lot of time, and it's just awkward to try and make work every time. And what I'm really excited about is actually this seems to be a way that we can just cut through all of that.